Yeah, so since I can think back, I have been really fascinated by life in all its forms. Like many of us, as a little kid, I was intrigued by the little fuzzy and furry animals. Oh, I need to push that button. And a little later, I was not only content with the fuzzy and furry ones, but learned about the wildest and weirdest creatures that you could find. And then still a little later, I read all I could read of pathogens and germs and parasites that infect and threaten our lives. And I was really scared by this. And as a teenager, I had a lot of time. At that time, we did not have internet and we didn't have mobile phones. So whenever I had time, I thought what it was actually about, that bizarre thing that created the human brain, and with the human brain, the ability to create symphonies and rockets. It's pretty amazing if you look at it, that we even can think about ourselves. And that's something that is intrinsic to life as a phenomenon. So I really wanted to know what is the definition of life. And if you look in textbooks, there's not really a very nice and satisfying definition. It's usually a selection of criteria. You have metabolism, you have replication, information. But what I found most intriguing about life is that although every single creature has only a short time to exist, the whole thing seems to be a never-ending and ever-progressing story. And even the smallest and simplest life form has the ability to learn and solve problems. So we humans take a lot of pride in our ability to solve problems, but if you look closely, even the smallest organism has this ability. And it was Darwin who basically framed an expression for it, evolution, almost 200 years ago, and we still don't fully understand evolution because it must have started with the constitutive elements of life, with molecules, because molecules were at the beginning of it. But when and how did molecules have the ability or come up with the ability to learn and solve problems? I couldn't imagine a more interesting question than that one. But then I did not end up studying biology, because in school I found biology really, really dreadful. So many facts to learn. The citric acid cycle and the endoplasmic reticulum, and I much more preferred physics. There was also a lot of facts that I didn't care about, accelerators and engines. But what I liked about physics was that physics education made it pretty clear that if you want to understand anything from scratch, you have to start doing the math. And I was pretty good at math. Well, I studied physics and I found that I got more math than I asked for. But what I really liked about my physics education was experimental physics, and particularly optics. Optics is a fantastic branch of physics because it's so visual. And you have these lasers and the optics, the optical table and the lenses and the mirrors, and you can basically build optical systems all by yourself. You don't have these huge teams and huge machines like in other branches of physics. And what is also pretty cool about optics is that you can, with optical instruments, study living objects. Really, the first microscope was invented to study biological specimen, almost 500 years ago. And that's how it looked like. And it was actually pretty good already. You could see the small structures of cork and even little microorganisms. During my PhD, I stayed with optics and I developed a method that was able to detect single molecules. And by a fluorescent label, so we could study not only where they were in a system, but we could basically study the dynamics. So I developed a technique that allowed us to very quantitatively analyze how molecules are interacting with each other. And if you will, that's the basics of all life, the interaction of molecules. So we measured interactions of proteins with DNA and other molecules, and we measured very precisely and understood these systems relatively well, but we measured in aqueous solution. And of course, our biological partners, they told us, well, okay, you understand something about molecules, but that's not life. You have to measure in cells if you really want to understand life. Okay, that's clear. So we moved to cells. Cells are transparent, so in principle you can apply your optical techniques also to cells. But what appeared to be first a technical problem, you have to get rid of the background noise and all that, very quickly turned into a very obnoxious problem, because the system that we wanted to study, that we could study very nicely in solution, in the cell was very hard to target. Every day we measured something else. So it took us really 4 years of preparing a specific cellular system in a zebrafish organism so that we in the end could make sense of what we measured somehow, right? The error bars of our curves were still such that it made every quantitative researcher cry, but our biological partners, they were quite happy with what we measured and made some sense of it. But at that time, I have actually witnessed the problem with biology. It's fascinating, but it's also super complex. Because a cell is not just 2 molecules, it's not 10 molecules, it's not 1,000 molecules. It's an incredible number of different molecules and an incredible number of interactions. If you will, you can compare it to the complexity of interactions in the internet, right? Good luck with understanding that. And even good luck with, looking at a single system and trying to control all the rest. It's not going to work because it is so complicated. And at that time, I decided, okay, so zebrafish is definitely not my organism. So I would like to have a system that is really simple, but still alive. Yeah, good luck. There is no such system, because a cell is composed of thousands and thousands of different components. And if you want to understand its essence, in principle, you have to reduce the complexity. But to try and reduce the complexity from the top, for example, by taking elements out of the cells, this is possible by genetic modification. You will not be able to really beat the intrinsic complexity significantly. So in fact, if you really want to start understanding a system that you can control from scratch, in principle, you have to build it. And that's a quote of a very famous physicist, Richard Feynman. I guess some of you have heard of the name. He basically, in a completely different context, says what I do not create, I do not understand. So in principle, if you really want to understand a system such as life, you have to create it. Of course, now we are a little bit in a shaky water here. Is it allowed to create life? We all have this idea of Frankenstein, and it's probably not what you should do. But in a way, we're looking at molecules that are coming together to do something to make the transition, in principle, from chemistry to biology. And that's far, far away from any Frankenstein. But how can you do it? We're coming back to our phrased problem from the beginning. Life is supposed to solve problems. So what do we have to do to bring molecules together such that these molecules are solving problems? Which problems are they supposed to solve in the first place? Well, to put it basically very bluntly, the first thing that these molecules need to do is somehow self-organize. There's a very, very important and very famous book by another physicist, Schrödinger, who went into exile in Ireland during the Second World War. And there he had to lecture, and he did not lecture about his Schrödinger equation, he lectured about what is life. It was not his home turf, but it was very interesting what he said about life. Some of what he said was quite wrong, but something was really, really cool. And that is that life is able to create order out of disorder. We all know about entropy. Everything wants to go to hell, right? Everything wants to mess up. I mean, we know that. If you look at your desk at home, you have the feeling entropy is acting all the time. And life is able to undo that, to sort molecules, to make structures and patterns, and even greater structures and patterns. And these develop into something like an organism that is very, very well controlled, that beats entropy all the time. And this is possible by the input of energy. Today, we have just had our energy, right? The earliest organisms probably used sunlight, we don't know. So we need something that is able to create energy, to self-organize. And we also need some sort of a box around it, a compartment, right? So every living system is compartmentalized. It has a very nice shell, and this shell should not be a box that with solid walls. It should be a boundary that is able to transform, because life wants to continue and replicate. So you need something that is keeping, the self intact, is able to basically keep things in and out, and at the same time should be easy to transform. And nature has found an amazing solution to this with phospholipid membranes. The shell of all your cells, of all cells that you can find on Earth, they all have phospholipid membranes. These are very thin layers of molecules that have a water-loving head group and an oily tail, and they basically form little double layers with the water-loving head groups outside and the oily tails inside. And they are keeping everything, every biological molecule inside that needs to be in, and keep everything out that needs to be out, and they're very transformative. So in my research, and that's now the last bit of my talk, I will show you a little bit of a glimpse of what we've already accomplished, because we need molecules that are self-organizing, but we also want molecules that are doing something. Self-organizing is fine, but they have this shell, and if they really want to live, they have to replicate, they have to divide the shell. So we are at the moment able to create a system of molecules that are self-organizing within a transformable shell and start changing the shape of this shell towards, not quite there yet, but towards the self-division. And I show you now a little movie where you can see it, and I'm not sure how much time we still have. Maybe you can see it twice. It's not easy to see here. So what you can see over time is that structures are forming. These are filaments made out of protein molecules. And these protein molecules, these filaments are basically able to transform the shape of our compartment, but they need to be arranged into the middle of the compartment because they need to divide it in two. And what you can see here very nicely over time is how these molecules first form the filaments, but also how they are basically, by an oscillation reaction made possible by another species of molecules, that are basically driving these filaments into the middle of the compartment. And what you can see here very beautifully, I think, is that they start constricting this volume. We are not there yet. The force to constrict this volume is not yet great enough, but we're very optimistic that we have the next factor in our hand to make that possible. And what you see here is a system that consists of a membrane and 5 proteins. Still 5, not 2, but it's pretty cool, I think. And of course, that is very far away from anything that lives, but it's the first step. And now I give you a homework. You don't have to do it, but we can basically talk about it in the speaker's corner afterwards. I want you to think about, once we have accomplished this division, and I think we're pretty close to it, what should we do next? What is the next important thing that you would like to see from a living, or a system that is supposed to be living at some point? Many things possible. Maybe you just think about it and let me know. What I think, it's very hard to say when we will be actually convinced that what we have in our hands under our microscope will be alive. My personal conviction is that, well, probably only if that thing really surprises us somehow. Thank you.